Hello students, welcome back to our channel, the Stephens Forensic Academy. So in the previous video, we have discussed the structure of DNA, the basic structure of DNA, role of DNA in the forensic science. Clear? So from the examination point of view of UGC net forensic science, this topic is the most important topic because in this from this topic we can see one or two question from this topic. Basically the structure of DNA and the history of DNA. History of DNA fingerprinting. Basically, we can say history of DNA fingerprinting are the two most important topics which can cover the other most important topics also, such that RFLP technique, PCR method, STRs, VNTRs, and another different extraction techniques. We can just discuss with the connection with these topic. Okay. So the medium of our language of this video will be Hindi plus English both. Okay, so both people of Hindi and English medium can understand this video. Clear? तो अगर हम बात करें कि डीएनए की हिस्ट्री के बारे में, इफ वी टॉकिंग अबाउट द हिस्ट्री ऑफ डीएनए, बेसिकली द हिस्ट्री ऑफ डीएनए स्टार्ट होता है हमारा 1985 से, when the Alex Jeffrey was the scientist who described the process of a whole process of DNA fingerprinting. Clear? So before starting this video, we have an announcement that if you want any access or if you have any query or a doubt about any means topic, you can like contact me personally anytime. Okay, from any social media account, that is Instagram, Telegram, or WhatsApp. Clear? So now we are going to start our video. First of all, you discuss about the history of DNA. Basically, history of DNA, the DNA fingerprinting was first described by Alex Jeffrey in 1985. Okay, so he used the some we can say repeated unit over and over again on the DNA, and that repeated unit are known as VNTR, that are variable number of tandem repeats, which are we can say vary from different from individual to the individual. Clear? So that VNTR reason, we can say technique called the RFLP technique. Okay, means we can say that that VNTRs are separated by a technique that is called the RFLP techniques. Okay, basically this is the restriction fragment length polymorphism technique. Okay, restriction fragment means, okay, this we can say, if we break down this name, okay, into different components, RFLP. R means a restriction. Okay, RF means a restriction. Restriction means that we have to use these some enzymes that are known as the restriction enzymes. Okay, that are known as the restriction enzymes, which we can say are known as the biological scissor. Okay, so with the help of these scissors, we have to cut the DNA into the fragments. Okay, we have to cut the DNA into the fragments. That's why known as restriction fragment. ओके, फ्रेगमेंट्स में हम इसको काटेंगे. ओके, then length polymorphism. Okay, length polymorphism means that we have to cut the different we can say the strand of DNA into different fragments that will show the polymorphism. Poly means many, morphism means form. Okay, that is the polymorphism. Okay, polymorphism means different forms of the DNA. Okay, different forms of the VNTR sequence which vary from individual to the individuals. Okay, and that reasons are known as the VNTRs. Okay, that reason are known as the VNTR, that are known as variable number of tandem repeat. Okay, tandem means again and again. Okay, which are repeatedly again and again, and with a different we can say sequence change. Okay, variation, or with we can say different sequence in the nucleotides, or with different we can say number of nucleotide present in the these VNTRs. Okay, clear? So this is just an introduction. Okay, if we know about the RFLP, we have the two type of technique. One is the SLP, second is the MLP. Okay, so SLP is the single locus probing method. Okay, SLP is the single locus, means we have just only one. We will go work with only one locus. Okay, we will only one locus. But in MLP, this is the multi-locus probe, which means that we have to work with multiple locus. We can just make the profile, or we can say work with the different locus. Okay, that is locus 1, locus 2, locus 3. But in single locus, we have to work with only one locus, and for the other we can say VNTR, clear. We have to strip off that region of the DNA from the nylon membrane, then we have to again bound with another sequence of the DNA. Clear? Is it clear now? Okay, now if we come to our main topic, that is the first of all we have to the main different eras of the DNA years. Okay, first era is the pre-DNA year. Okay, first era is the pre-DNA. Okay, that is from 1900 to 1935. Clear? Second is the first decade of DNA. Second is the first decade of DNA. Okay, that will be from 1935 we can say in 85, then from 85 to further 200 to 2000. Okay, we have the second decade of the DNA. Clear? Is it clear now? Okay, now prior to the DNA, the what are the different methods in the forensic we will use? Okay, we were used. Okay, so there was the different we can say techniques such as the blood grouping techniques and the protein profiling we used before the DNA invention. Clear? Or DNA fingerprinting. So in 1900 to 1985, there were blood grouping. First of all, we have blood grouping testing. In 1900, the Karl Steiner was a scientist who described the different we can say blood groups. Okay, different blood groups described by him, and that named as AB, ABO, AB, AB and O blood group. So there are four type of blood groups, and for that he got the Nobel Prize in 1930 in the medicine. Okay, so there are different proportion, different we can say population have different proportions. O has 43%, A has 42%, B has 12%, AB has 3% of we can say blood group. So blood groups are related to the antigen polymorphism. Okay, now what are the blood groups? So how we can say it is of A blood group, it is of B blood, it is of O blood group. Okay, so it depend upon the different type. We have a RBC. Okay, so on the surface of RBC they have different antigen polymorphism. Okay, on the surface of RBC they have different antigen polymorphism. So the antigen which is present on the surface of RBC, which is present on the surface of RBCs. Okay, so that RBC will demonstrate that blood group. Okay, that RBC will we can say demonstrate that blood group. If we can say that if it has A antigen, that is of A blood group, it will have B, then it will have, if it will have both A and B antigen, it will offer both A and B blood group. Okay, if will not have any antigen, then it will go for O blood group. Clear? Clear? So first was described like this. So then after that the Leon Lattice was the first scientist who described the we can say blood grouping techniques of blood typing from the dried blood stain. Okay, with the antibody test for the AB blood groups, and first used this data in the Italian court around 1915. Okay, so after that AB testing is used in the paternity dispute in the Europe and United States. Okay, after the AB blood group, we have another different blood group system, that is the MN system. Okay, MN system is further discovered in the 1927. Okay, and after that we have a different blood group system, that is the RH factor. Okay, on the basis of we have the blood group A positive, A negative, okay, B positive, B negative. So that positive and negative will be represented by the RH factor. Okay, so Alexander Wiener and Leonard Steiner discovered RH factor in 1937. Okay, 1937, the Alexander Wiener and Leonard Steiner discovered one more factor, that is the RH factor, when they were just working with the rhesus monkey. Okay, so individuals with classified as RH positive or RH negative. Okay, so in the forensic science we have to deal with the serum. Okay, we have to deal with the body fluids. Okay, so that come under the one section that is known as serology. Okay, that is called serology. Or we can say that serology is a term which is used to describe the laboratory test that examine the specific antigen antibody interaction. Okay, so this is the table which is showing the we can say inheritance of the blood group, different blood groups. Okay, so after that, with the advancement of techniques, we have a different technique that is called forensic protein profiling. Okay, that is the forensic protein profiling. So all we know that the proteins are made up of the different amino acids. Okay, different amino acid sequences. Okay, so these sequences can vary from individual to individual. We can say 1 in 100. Okay, one sequence is present in only 1 in 100. We can say. Okay, so they will show the polymorphism in different forms. They will show the different forms in different individual. So we can say we can discriminate these proteins on the behalf of their profiling, okay, different profiling. So this will, we can say, lead to a very best application in the forensic science. Clear? So in a protein profiling we have different isoenzymes, okay, we have different isoenzymes. Okay, now what are the isoenzymes? So isoenzyme, we can say that enzymes are all the proteins, okay. Iso means same, okay. They have the some different, we can say they have some same proteins called in protein sequence of amino acid, but vary within the different individuals, okay. Clear? So here a technique that is the, we can say, forensic protein profiling. Amino acid sequence in the some proteins vary among individuals in the human population. The use of multiple protein polymorphism can lead to the modest power of discrimination, where the chance of inclusion is 1 in several hundreds. Clear? So the human RBCs, okay, the human RBC, the serum, basically the serum contain the all protein content. Okay, so the serum contain different isoenzymes. These are the multiple forms of proteins present that can be catalyzed the same biochemical reaction in spite of the slightly different amino acid sequence. Okay, there is slightly difference in the amino acid sequence, but they can perform the specific function. Typically, 2 or 3 forms of each isoenzyme. The techniques which we used for the separation are the starch gel electrophoresis, agrogel electrophoresis, polyacrylamide gel electrophoresis was used early in the, we can say, separate these protein into distinguishable alleles. Okay, so by 1980, many labs used, now here when the year was 1980, so the isoelectric foxing technique was used. The combination of the, that is the isoelectric foxing polyacrylamide gel electrophoresis, having the combination with the silver staining technique. Okay, silver staining technique, okay, which we like used in the further more techniques in the STR. Okay, so here the we use the silver staining, okay, we use the silver staining technique. Okay, so there are different, we can say, erythrocyte isoenzyme present in this list, such as phosphoglucomutase, okay, that is PGM, ACP, ESD, AK, GLC, AD. Okay, there are different number of alleles are present. Okay, there are number of alleles are present, and there are different type of discrimination potential, okay, present. Clear? After that, when that was the first, we can say, era of the, we can say, prior to the DNA. So in 1935, we can say, or 1909, sorry, we can say 1985. Okay, that was the, I think, just to make some correction, that was the 1900 to 1985. Clear? So next जो first जो decade होएगा हमारा, that will be from 19 first decade 1985 to 1995. Okay, so please make correction there. Okay, that was 1900 to 1985 and 1985 to 1995. Okay, first decade means 10 year. Okay, decade means 10 years. Clear? So Alex Jeffrey 1985 work with the VNTR using the RFLP technique. Next, the Wayman and White shown that RFLP marker has show the polymorphic. Okay, so they show the Mendelian inheritance. Okay, there in the for upcoming generations. Okay, so I think all you know about the Mendelian inheritance. Okay, that is part of genetics. So the result with the Jeffrey multi-locus probe looked similar to the barcode. Okay, I think you all know about the barcodes. So when they just perform the multi-locus probing technique, the result were like that of the barcodes. So the patterns is called DNA fingerprint because they are like fingerprints are unique to each other. Okay, like our fingerprints are unique to each other. So that barcodes are also, we can say, unique to each other. So that's why they are called the DNA fingerprinting. Okay, because the variables are unique and gives the unique pattern to each individual. Jeffrey teamed up with the Patrigal and David Rett published his first forensic application of DNA fingerprint in December 1985. Okay, and then at the same time, the Carey Mullis, okay, who was the inventor of the PCR, okay, invented the PCR to make the copy of any desired region of DNA. Okay, so PCR is a further another technique that is polymerase chain reaction. Okay, that was a technique to like make multiple copies of a desired region of the DNA. Clear? So first of all, if we discuss about the RFLP technique, that is the restriction fragment length polymorphism. Okay, so just pay attention here. We are going to discuss this technique, okay, in detail. So first of all, you discuss about the RFLP technique. So this technique, we can say, have different steps. Okay, this technique had different steps. Okay, clear? So the steps are like that. First of all, we have to extract the DNA. Okay, first of all we have to extract the DNA. Extracted DNA means DNA different. We have different extraction techniques are there. Okay. After that, we have to after the extraction of DNA, we have to cut the DNA into different fragments. And after the fragmentation, we have to just separate the different fragment on the gel electrophoresis. Okay. Then we have to transfer the DNA fragments on the using on the nylon sheath using the southern blotting technique. Okay. Then hybridization, we have to hybridize the locus specific probe, then a detection of locus band with the autoradiography or chemiluminescence techniques. Clear? So if we discuss here, so this techniques took several weeks. Okay, first of all, the first limitation is that this technique you take the different weeks. Okay, multiple weeks. Okay, for the completion. So that time there was no other option. Okay, now we have the PCR method, but that is there no other option there. That's why we use this technique, RFLP technique. Okay, so this technique you took the several weeks to complete. First of all, we have to take the sample. Okay, collect the sample such as blood or other biological material. Then we have to cellulize, or then we have to extract the sample to break the cell membrane and removing the package of protein and around the DNA. That is the cell extraction. Then the addition of restriction enzymes. The restriction enzymes are biological seizure that are capable to finding the or cutting a specific DNA sequence. Okay, that extracted DNA. Clear? After that, after that, after that we have in the restriction enzyme we mostly use the HAE3 enzyme. Okay, that is the come from the Haemophilus aegypticus. Okay, that is the bacteria. Okay, that is name of bacteria, that is Haemophilus aegypticus, which recognize the GC rich region. Okay, where the GC rich region is present, then that will cut, or we can say cleave the DNA fragment. Okay, and form the sticky end. Okay, that sticky end known as the palindromic sequence. Okay, so that question can be like that the what the sticky ends are known as. Okay, that are the palindromic sequences. Then the chopped DNA fragment then separated by the fragment size on the regular gel electrophoresis. Okay, then we have to perform the electrophoresis. Clear? After that, we have to do the southern blotting technique, that is named after the scientist, that is Sir Edwin Southern. Okay, 1970. Then we have to separate the DNA fragment were transferred from gel to the solid matrix. Okay, then we have to just then we have to separate the DNA fragment. Then we have to transfer on the nylon membrane using the different technique under the alkaline condition. Okay, by using the NaOH. Okay, that is sodium hydroxide. Then DNA strands were we can say reduced to single strand. Okay, here the denaturation take place. Okay, the DNA strands now there are two strands now get denatured. Then the DNA will be immobilized on the nylon membrane using the UV cross-linking process. Okay, the one strand of DNA fixed to the membrane by cross-linking the DNA onto the membrane by using the UV light. Then we have to hybridize that DNA using the radioactive probe. Okay, then we will use the radioactive probe. The VNTR the VNTR radioactive probes are allowed to hybridize to the DNA under the optimal temperature and ionic strength, and labeled probe find out their complementary sequences. Clear? Then we have to washing out the excess of probe from the nylon membrane. Then we have to detect the different fragments, or different we can say profiling of the DNA profiling we can analyze on the nylon sheet. Okay, by using the X-ray film. Okay, so for the detection of a probe we can use the two type of technique we will discuss further on okay in this video: radioactive labeling and enzyme conjugated probe. Okay, then a formation of a autoradiogram. Just remember this is called the autoradiogram. Okay, so the all barcode like structure will be formed here. Then we have to compare with it the standard sample. Okay, clear? The covered through the scanning and comparing with the band position. Clear? So after that, to detect the additional VNTR. Okay, first of all, if we have done the first cycle of the VNTR, first cycle of the RFLP, if we want to detect the additional VNTR, then what we have to do? Then the membrane would be stripped off with the alkaline solution, and then we have to rehybridize. Okay, we have to do again rehybridize of the membrane take place. Clear? So one thing more is that in the RFLP technique we have to like we can say discriminate, or we have to determine by the bins. Okay, not by the discrete allele. Okay, in RFLP technique we have to use the bins. Now what are the bins here? Bins are the DNA fragment that differ from only with a few repeat unit. Okay, but in PCR we use the discrete allele. Okay, we have to use the discrete allele for the discrimination, But in RFLP technique, we have to use the WINs. Okay, WINs are the small DNA fragments, or we can say DNA fragment or sequence. Okay, so this technique take some several days to the weeks. Okay, so there are technique that is multi-locus probe. Okay, so multi-locus probe can detect multiple VNT loci in simultaneously. Okay, simultaneously we can just detect the different loci. Clear? So we have the different locus, locus A, locus B, locus C. Clear? So further, if we take some VNTR or GC reaches sequence 10 to 15, this core sequence hybridized to multiple VNTR, then share these core sequence. As a result, MLP produce a complex barcode like a band pattern from allele of multiple VNTR loci. So MLP technique was called DNA fingerprinting. Clear? So this is explaining the DNA fingerprinting. Now there are advantages. So advantage is like that a high power of discrimination. Second is the method was used for the parentage testing for immigration dispute with the greatest success. So the disadvantage is that this interpretation of mixed. Okay, if we have a mixed DNA sample, then we can't use this technique. Okay, the basic disadvantage is that the interpretation of mixed DNA sample for more than one individual is nearly impossible due to the complex DNA fingerprint pattern. Clear? So this is the, we can say, diagrammatic view of the overall RFLP technique. Okay, so here we have the restriction enzyme. We have to cut the restriction enzyme on gel electrophoresis. Then these are the different enzymes, that is HAE3, okay, HINF, okay, PSF1. Okay, so these are the different enzymes, restriction enzymes. And then we have to, we can say, fix on the agro. We have to just separate the using the agrogel electrophoresis. Then we have to denature under the alkaline condition. Then we have to transfer using the different technique, that is southern blotting, okay, on the nylon membrane by using the UV cross-linking. Then after that we have to detect the all the DNA sequence like a barcode. Clear? So we have different technique, that is single locus probe technique and the multi locus probe. Okay, single locus probe we have already discussed. Okay, just read here. Now what are the restriction enzymes? So restriction enzymes are proteins, okay, discovered in the various bacteria. What are the restriction enzymes? Restriction are the proteins that is discovered in the various bacteria. That formation as that is biological seizure cutting the DNA molecule precisely at a specific C pattern. Okay, specific pattern में उसको काटेंगे. Okay, that will lead to the sticky ends. That ends are known as the palindromic sequences. Okay, because they read the same in both direction. Clear? Because they read the both direction. For example, these are the some sequence. If this is the 3 end, this is the 5 end. So this is the 5 end, this is the 3 end. Okay, so if we say that this is the G, C, G, just taking an example. So this is also the G, C, G. Okay, so like we can say Nitin. Okay, you can read this name from for backward or forward, and there will show the same represent. Okay, so that is known as the palindromic sequences. Okay, this is example, that name and Nitin is the example. Okay, so restriction enzymes can cut both strands of DNA, generate either sticky end which have an overhead of few nucleotides. Okay, so they have further some nucleotides, okay, present, okay, excess, so that will like help to bind the radioactive probe. Restriction and nucleus cleave the phosphodiester bond of the DNA. Okay, I think you know about the phosphodiester, we have discussed in the previous video, and a specific site known as the restriction site. The we can say these type of restriction enzymes on the basis of subunit composition of enzymatic properties. So there are three type of restriction enzymes, basically type 1, type 2 and type 3. What we use the type 2, type 2 is the most important, commonly used for the require magnesium as a cofactor. Okay, in this we have to use the magnesium as the cofactor. So here the HA3, that is HAE3, that is the come from the Haemophilus aegypticus, and 3 stands from the third endonuclease isolated from that bacteria. Okay, now if we go, if we have to detect the we can say X-ray film. Okay, we have to detect the last product of the RFLP. Then we have used two different techniques. Okay, one is the radioactive labeling. Okay, second is the enzyme conjugated probe. The radioactive labeling जो होएगी हमारी, okay, the radioactive labeling, that is we use here isotope of phosphorus, that is 32. Okay, we have to use the 32. Okay, probe can be used. The hybridized probe can be detected by exposing the membrane to sheet the X-ray field to generate the autoradiograph. Okay, and further technique is like that. Alternatively, we can use the enzyme conjugated probe can be used in this alkaline phosphatase. Okay, this is the enzyme. Alkaline phosphatase enzyme is used. Its enzymatic activity can be detected by chemiluminescence. Okay, this process is used in the chemiluminescence substrate. A chemiluminescence signal can be detected by exposing the X-ray film as well. Okay, clear. So we have further technique here is the longer range PCR. Okay, now we are discussing about the RFLP. Okay, so once the RFLP technique has been discovered, so once the different application has been done with the RFLP technique, now we have to come with a further another advanced technique, that is the PCR. Okay, so in the year of, we can say, 1995, there was the experiment was done on the PCR method. Okay, so in the late 1990s, okay, in the late of 1990s, an attempt was made to develop the ability to PCR amplification to entire VNTR region to improve. Okay, means to improve the result of RFLP, we just using the PCR method just to amplify the sequence of VNTR. Okay, so we just using the long PCR for to increase the sensitivity of that technique. Okay, so for that we here are using the one more enzyme for the amplification, that is the tag polymerase. So tag polymerase is the thermostable enzyme. Okay, thermostable enzyme. Clear, we will discuss in the PCR method. Okay, this further. So there are early PCR based testing. Okay, before the PCR there are some early PCR based kits, or we basically we can say that PCR based DNA testing techniques was used, such as DQA1, D1S80, a PCR amplified VNTR, PM plus DQA1. This is the combination. Okay, and second, third, fourth are the STR. Okay, that is short tandem repeat. So first basically use, that is the HLA, that is the human leukocyte antigen. HLA is used in the paternity. Okay, dispute में हम use करते हैं HLA को, that are used in the paternity cases. HLA, that are the human leukocyte antigens. DQ alpha. Okay, DQ alpha, the first PCR based DNA test kit for detecting the sequence variation in the HLA DQA1, that is alpha 1 gene, found on the chromosome number 6. Okay, this is the chromosome number 6, we have found the HLA. Okay, remember this. Okay, sometime in the like exam you have to ask about the chromosome, number of chromosome, or we can say HLA sequence. Okay, clear. So the Citus Corporation released the DQ alpha ampli type kit in 1990. Clear. So further, with the discovery of PCR, we just have to discover, there is some more discovery were done, that is of these short tandem repeat. Okay, STR markers were found at that time in the year of 1980s or 1990s. Clear. So STR markers are have different application over the VNTR. Sorry. So STR markers in late 1980s at about several time of D1S80 other mini satellites. Okay, one thing more, VNTRs, VNTRs are known as mini satellite, just remember this. And STR markers, that is short and repeat, are known as the micro satellites. Okay, just remember this, micro satellites. Okay, these satellites basically on the chromosome we have a structure, more extra structure, we can say extra structure, that is called the satellite. Okay, then only satellite DNA, also known as satellite DNA. So this, remember this, because in the December 23 we have we have a question like that, the microsatellite is also known as. Okay, like that, they have a question, so remember this. So microsatellites or STR were also reported having 2 to 7 base pair. Okay, just remember this. The VNTRs are the VNTRs are how many base pair? VNTRs are 10 to 15 base pairs. Okay, 10 to 15 base pair longer repeated sequences. What STR markers are the 2 to 7 base pair sequences. Clear? Clear? The STR markers were being widely used and would be genetic marker of the choice for the future. Okay, so STR marker have very, we can say, advantage over the VNTR. Okay, so STR marker have some advantage, such as STR marker can be amplified by using PCR. Okay, what VNTRs we can't use in the PCR, STR markers can be amplified using a PCR because of their small size. high rate of sensitivity. STR markers have high variable number. Okay, variability इसमें ज़्यादा होगी. Okay, STR marker have a high variability. So in 1995, yeah, UK established the first database. Just remember this: the UK established the first database that is known as CODIS. Okay, that is known as CODIS. CODIS, the first database of the DNA. Okay, that is the combined DNA index system. Clear? The for criminal investigation, US first selected 13. Okay, US selected the first 13 STR loci. Okay, the first 13 STR loci that are known as combined DNA index system, that is called CODIS. The first commercial STR kit capable of multiplex amplification became available for the from. So their first kit was we can say available from the Promega Corporation 1944, in which we are still using the three STR loci that are the SCSF1PO, TPOX and TH01 that are named as CTT. Okay, that are named as CTT. Clear. So in this the syllabus training analysis was done. Okay, so this is the structure we can say representing the STR marker or VNTR marker. This is the mini satellite, that is the VNTR. Okay, this is the D1S80. Okay, this is the flanking region. Okay, this is the sequence of the this marker. Okay, and this is the TH01. Okay, only both 4 base pair. This is the 16 base pair, and this is the only 4 base pair. Okay, TH01. Clear? So there was second decade, that is 1995 to 2000. Okay. So here the working was done on the in the 2000. I think there was human genome project was we can say initiated. Okay, so now human genome project is also again in the news. Okay, so in this time there was different we can say markers are used for the distinguishing in the sexual offences case, or we can say for the paternity case. Okay, so here the we can say there's different sequencing techniques are also evolved. Okay, in 1997, that is Sanger, that is Frederick Sanger, the Sanger sequencing was also done. Okay, then in the same sequence, that same like that of if you have to extract the DNA. Okay, the processes will be same. Okay, here the difference is that we have to use the DDNTPs. Okay, that is the D-deoxy. Okay, D-deoxy nucleotide triphosphate as a chain termination. Okay, the other process is same. Okay, so there are the mitochondria DNA now come into news. Okay, in this era, mitochondrial DNA. Okay, if you know about the mitochondrial DNA, please watch my previous video on the mitochondrial DNA. Okay, mitochondrial DNA, which is very helpful in the paternity cases. Okay, paternity and maternity cases is very helpful. Okay, basically this is mitochondrial DNA is used in the maternity case. Okay, mother linked DNA, because a mitochondrial DNA will transfer from the mother to the child. Okay, then we have the bichromosome. Okay, bichromosome. Okay, bichromosome inheritance also done here. Now we have a different further one more gene, that the amelogenin. Okay, amelogenin come into news in this era. Okay, so for Y chromosome and amelogenin we have the different separate video. Okay, we are coming with different separate video. Okay, now for today's we have just discovered only these we can say history of DNA. Okay, so for the next video we will discuss the different extraction technique of the DNA. Okay, and this unit of a UGC net forensic is almost covered. Okay, if any topic is left, we will discuss the further. Okay, like a short video, like 5 to 10 minute video, we will make that. Okay, clear? So that was all about for today's video. Okay, if you have any doubt regarding this, or if you want any type of notes or handwritten, or we can say computerized form, then you can ask me anytime. Okay, or any social media platform. Okay, so that is all about today for today's session. Thank you so much.